Phenol red, here's some clues. The Durham tube. The only experiment we did with Durham tube is phenol red. The other thing is start to look at the cap colors. The cap colors are very different as you go across, right? The only other tube that can possibly have a red cap is this one. But look at that, that's a solid. So that must be what experiment? It's solid. TSI. The one with a liquid with a Durham tube is what test?
Phenol red.
Phenol red. Glucose is the red one, lactose is the green one. All right? But the idea is if you know the experiment, it's going to be easier to interpret. If I look at urease, urease is the only one with the green cap on it. And it's a small one. Everyone see that? And it's a pink color. The only one with blue caps now. If you look at them, we have litmus milk. That looks very different. We have gelatinase. Can be solid or liquid, right? And then we have MR, VP, which are red in color. So the three blue cap ones are all very different from one another. So use that as a guide to tell you which experiment you're interpreting. All right, that makes sense? So if you look at the blue cap, you look at the blue cap ones. You see all the blue cap ones are different. So MR,
number 4.
litmus milk, gelatin. So start to learn the cap colors that give you a clue to what test it is. All right? You got a half hour. Red solid, TSI. TSI, and then you could have all different types, but
That's the starch plate. That's the starch plate.
Solid media, that's
So what's the difference between both? Professor, what is the difference with these two? Why the starch plate? This was 2 organisms, this is 3 organisms.
They just got some 3 organisms on the plate.
Okay.
But the idea is that iodine binds the starch.
Yes.
So you can see there's no starch around that cell because there's no binding. So what enzyme must have been secreted by that cell? What's the name of the enzyme that hydrolyzes starch?
Lipase?
Not lipase, lipase is lipids.
It's lipids, yeah.
Amylase, like salivary, like we have amylase, right? So it secreted amylase, broke down the starch, therefore iodine cannot bind with it. You could see this organism did not produce amylase because there's still starch all around it. So when you see clearing, the interpretation is amylase was
present.
Just by that color, which one it is. You know what I do? I look on top of that white color. When it is gray on top of that white color, that's reduction. So when it's gray on top, white on the bottom, that's reduction. When I see pink on top of that, that's acid production. Because pink is acid. If it's all white, like if it's, you ever see milk do this in your fridge? That's acid with gas. That's what we call storming fermentation. You produce so much acid, you see all that solid went towards the bottom, and all the liquid there is at the top. It's separated.
So this one would be acid?
That's acid, but that's acid with gas fusions. Yep. Call that stormy fermentation. So the key is, it's never going to be all white. Not going to be all white. Look at the second layer to distinguish between acid, clot, and reduction.
Pink on top, white on the bottom. Then would be
So acid clot. And that's actually a clot, because when you look at it, it's actually solid.
If it wasn't solid.
It would still be acid clot. The clot hasn't formed. Listen, these are the seeds on our exam.
Okay.
You know what I mean? Unless someone ruins it.
Sorry.
No, that's a black.
The organism is what? Pseudomonas. Pseudomonas is green. So what you're seeing there are all the green cells. Really what digestion is it's brown throughout and it's clear. What I mean by that is if you look at the milk,
Oh, that's digestion, right?
that's digestion. You see the brown towards the bottom there?
Yes.
The key here is if you look at milk, light never passes through. Like I can't see light coming through. When digestion occurs, light passes through. The other thing is if you know Pseudomonas, which is green, digests, just remember that one thing, write down your book. Pseudomonas cause a color change. That means your organism cannot follow that pathway because no acetone is present. So you're testing for this intermediate called acetone. When you add your reagent, it turns the tea red. You have to memorize that whole pathway? No. But the idea is that some organisms can get energy from that pathway, other organisms cannot. Acetone, carbohydrate what?
Fermentation.
Carbohydrate fermentation. What are the sugars that are in here? What are the carbohydrates?
Glucose.
Glucose, lactose,
and
sucrose. Okay? So that's one thing you test for. What's the other thing we test for?
Sulfur reduction.
Sulfur reduction. Okay? So now let me pull up a tube here. Now I see, yeah, look, two parts we interpret, right? The butt area where it's anaerobic, and then the slant where it's aerobic. I see yellow in the butt, yellow in the slant.
Total fermentation.
Total fermentation of which sugars?
Glucose.
Glucose.
No, all of them.
All of them.
First with the glucose.
All of them, because here's how this is set up. We have very little glucose in here. We have a lot more lactose and sucrose. So here's what happens. Glucose is what organisms prefer. That's the first sugar it's going to burn through. Well, at some point, you're going to run out of glucose because there's not that much of it. So you have two choices. Either you can turn to lactose to ferment if you can do it, or you're going to turn to protein. The fact that it's yellow throughout means once we ran out of glucose, we use the other sugars for energy. Because when you ferment, what are some byproducts of fermentation? Acids are produced and gas. What gas is produced?
CO2.
Carbon dioxide. See the CO2 right there?
Yeah.
See the auger lifting? That's CO2. The pH indicator turns yellow under acidic conditions. So it must be fermentation of all sugars.